All right, this is going to be a quick little review of your notes that I sent out. You have your worksheets that you're working on too. That's over RNA and RNA transcription. So last chapter was on DNA, and DNA was that double-stranded double helix. And the DNA strand is not allowed to leave the nucleus, which we talked about way back last semester. It is contained in the nucleus. And if the DNA has all the information that codes for whatever that organism is, there has to be a way of getting that information out in those eukaryotic cells to the rest of the cells. Well, the way we do that is we actually change it into RNA. RNA is a little bit different. It does it is a nucleic acid, but it's able to get through the nuclear membrane. And it does this in three ways. Okay? The first one is the differences in these. So we'll just call them differences. One is the sugar is not a deoxyribose, it's just a ribose. So we have a difference in the sugar. So that's going to be a different chemical structure, different chemical imprint. And then we are also going to have, the second thing is that it is made up of a single strand. So structurally, it looks way different. Again, it's going to be a corkscrew shaped strand, but it's just going to be a single. It's like part of a ladder, half of the ladder that we talked about before, half of the spiral staircase. And number three, and again, when you transcribe these into your notes, you can go back and put a big star next to this one. We have an introduction to a fifth nitrogen base. Because again, even with a change in the sugar, a change in the structure, there is one other change that allows us to get out of the nucleus, and that in RNA, thiamine, T, is replaced by the nitrogen base U, uracil. Uracil, make sure you spelled that right. Oh, forgot my uracil. Okay, there we go, just so I can spell it right. So thiamine, the T, is replaced by uracil. So if we go back over to DNA real quick, we had that base pairing rule of A always binds to T and G always binds to C. Well, when we deal with RNA, when we transcribe this, when we make a copy of this, okay, whenever we come up to an A on our DNA strand, we're going to get an A will always bind to a U, and G always binds to a C, and again, vice versa. But this here is a bigger change. So when you're doing a test, a quiz, something in the future, and it talks about, hey, uracil is part of the nucleic acid that you're dealing with, the nucleic acid, you know you're talking about an RNA strand, a single strand that has in its backbone a ribose. Okay? So again, the makeup is the same. It has a phosphate group, a sugar group, and a nitrogen base. Differences are the sugar is a ribose, it's a single strand, and instead of T's, we have the nitrogen-based uracil. Okay, so we have a new base pairing rule too. Okay, now again, something else about RNA that is not similar to DNA is that RNA comes in three types. Okay, so just keep that up there. And there are three types of RNA. DNA only comes in one type. RNA comes in three. We have mRNA, which is messenger. I think that's supposed to be an E, not a A. RNA. And this is the one that actually works inside the nucleus. This is the one with the RNA polymerases, when we talk about transcription, that actually are copied right off of our DNA strand. So, just like we saw with DNA when we make copies to make our chromosomes, DNA gets opened up. So we have our DNA strand, and all of a sudden it gets this big wide spot in it right here. This is where our messenger RNA strand can come in, and let's do that with a different color. the messenger RNA comes in, an RNA polymerase will attach to it, and as it builds, as it goes this direction, it'll build a single strand of RNA. This is our messenger RNA. And this is the one that can leave the cell. Okay, so this can actually take the coded sequence, the codons that are on the DNA, and take it out of the cell and take it to where it's needed in the cell to either make hormones or repair cell parts or grow or whatever it needs to do. Okay, so we got our messenger RNA. It transcribes DNA into RNA. And, you know, we could add that to your notes. Maybe we can say it takes DNA and turns it into RNA. We can simply say that's what it does. Okay? Actually, and then we can put a little M there and say, well, it's actually mRNA because we have 3 different kinds. Now, the transcription process is something that we're going to touch on here in a little bit again, but I want to hit the other 2 types of RNA that we deal with. And the next one is tRNA, which is called transfer RNA. And this one is a translator. Okay? Now, if this is a transcriber, we got a transcriber in the messenger RNA and a translator in the tRNA. A transcriber is a difference in dialects, is how I usually say it in class. So if you live on the East Coast and you want to have a fizzy drink, you say, I want a soda. But if you live on the West Coast or in the western part of the States, we always say a pop. Now, you may have family members that some of them say soda and some of them say pop. Some of them may be from the East Coast. Some of them may be from the West. That's called transcription. There's a slight change, but everybody understands what you're talking about. Okay? There's a slight change in the sequencing. Other times I can use, Southern drawl versus an Eastern dialect or a Western dialect, where that comes into play too. But that's what a transcriber is. It's the same language. It's still nucleic acid to nucleic acid. Okay? But here we have a translator. What does a translator do? Well, just like what a translator does in any other situation that you can think of. It takes one language and turns it into another language. tRNA's job is to take nucleic acid language, and in this case, RNA language, and it's going to change it into proteins. Okay, and these are the building blocks of life. These are our enzymes and everything else. And it does this because it has its own special shape. tRNA actually does have kind of a T shape. It has this kind of weird key sort of structure. Okay, and it's still a single strand, but again, it's not just a single, like a piece of string. This one actually has this structure to it. Okay? This actually has a structure to it. And it does kind of look like a key. And it is attached to, on one side it's an amino acid, because, hey, we know that amino acids build proteins. And then it has this special portion on it down here, which we'll talk about more in our next video, called the and I spelled that wrong, the anticodon. Now, codons, as we learned in the last chapter, were sets of three nitrogen bases that code for some information. We know that our DNA strand is made up of introns and exons. Introns contain the information. Exons are spaces that allow for change and allow for things to evolve and go over time. So, when the messenger RNA reads this little RNA polymerase is going to cut out all of the exons and just make an information highway, an information string. This is only codons that code for amino acids. Okay, specific amino acids, 20 of them that we deal with. Okay, so proteins are built from 20 amino acids. 20 amino acids make over 100,000 different kinds of proteins. And again, here's that number that we've had in tests before. We have 4 nitrogen bases from DNA. They code for 20 amino acids in tRNA, and they make our 100,000 proteins, which build basically everything that we see on this planet. It's kind of a neat little thing. So we got messenger RNA, so we can get the messenger out. We have a translator RNA, which connects 2 languages together, a nucleic acid language to an amino acid language. The amino acid language gets put into proteins. The proteins make what we need. Now, how do these two things work together? Well, they work at that special little site in our cells, and we used to always draw it as a little dot. We just made a little dot. And then I said, well, it's a little more structured than that. It's actually kind of a double dot. It has two sides to it. It's got a protein side, and I'm going to just use blue again because it's RNA. It has an RNA side. And when we draw it really big, making it way bigger than it normally is, this was our good old ribosome. Okay, and the ribosome is where we make proteins. And the RNA is called ribosomal RNA. And this is a structural RNA molecule. It actually is part of the structure. And that structure is right there. Okay? And it is a binding site for our two other RNAs. The messenger RNA is going to attach right between the two. It comes in, feeds right in like this. Okay? And our tRNAs are going to bind to two sites up on here, okay, that help build our protein molecule. And again, we're going to go into more detail on that on our next video. But this is the three types of RNA. We have a ribosomal RNA, which is going to help with protein production. Okay, this actually is the site where we put together our nucleic acid, Na language, and our protein, our proteins. We can just put it that way too. This is what's going to happen right here. So those little ribosomes that we find in all kinds of cells, whether eukaryotic or prokaryotic, floating around in the cytoplasm, attached to the rough ER. This is where we make our proteins. They have to come from our DNA. They're going to get transcribed into messenger RNA. Okay? So this transcription process, and your textbook has a great diagram of it. It's on page 364. So again, you should have your textbook with you. Make sure you look at page 364 to get a better understanding of that process. And then in your notes, those bullets underneath the transcription part in your notes, I have, 4 of them there. Read through that. And it tells you that we make a copy of our DNA. The polymerase, in this time it's an RNA polymerase, it cuts out the exons, it puts the introns together, and it makes our messenger RNA that turns into tRNA at the ribosome, and we get our proteins at the end. And that's our introduction to the RNA molecule. Keep working on your assignment.